batches of B.Pharm graduates and 14 batches of postgraduate students and 8 batches of pharmacy students. Sri Padmavati School of Pharmacy has a tradition of enriching knowledge on current aspects of academics and research by organizing various scientific sessions, national and international seminars and symposiums on campus, and recently national and international webinars online and guest lectures, scientific sessions every year. In continuation of this tradition, we are organizing a 2 days national webinar on biopharmaceutical principles and validation in pharmaceutical industry on 8th and 9th of this month. In this session, in this webinar, we'll witness scientific sessions on current emerging topics by some of the eminent speakers in the area of pharmaceutical sciences. Firstly, let us start today's 2 days national webinar with scientific session by Prof.R.Nagaraju sir on a topic entitled Application of Pharmacokinetic Principles in Clinical Situation, which is an emerging topic for all the research scholars, staff, and students, irrespective of their specializations. Okay, and but before starting today's session, let me take an opportunity to invite dignitaries to introduce and invite dignitaries for today's webinar. Firstly, uh, it's my honor to invite Chief Patron for today's webinar, Srimati P. Sulochana Madam Garu, Chairperson of Sri Padmavati Educational Institutions. And patron for today's webinar, Sri P. Pranit Sir, Director of Sri Padmavati Educational Institutions. And convener for today's webinar, Professor Dr. D. Ranganaiklu Sir, Principal of Sri Padmavati School of Pharmacy and President of Indian Pharmaceutical Association, Tirupati Local Branch. And organizing secretary for today's webinar, Dr. V. Shanmugam sir, Head of Pharmaceutics Department, Sri Padmavati School of Pharmacy. I invite heartfully all the dignitaries and a special welcome for today's honorable speaker, Professor R. Nagaraju sir, Dean of Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalaya. Welcome sir, we welcome you.
Thank you.
Yes. And so welcome to all the dignitaries and let us start today's webinar traditionally with an invocation song. Jamal sir, I request to play the invocation song.
Swati namastubhyam varade kamarupini vidyarambham karishyami siddhirbhavatu me sada सिद्धिर्भवतु मे सदा सिद्धिर्भवतु मे सदा.
Thank you, Jamal sir. Now I request convener for today's webinar and principal of Sri Padmavati School of Pharmacy, Professor Dr. D. Ranganaiklu sir, to present welcome note for today's webinar. Ranganaiklu sir, I request you to please deliver welcome note.
Mr. Murthy.
Sir. Sir, it is audible, sir.
Is it audible?
Yes, sir, it is audible.
So, respected today's Chief Patron, Madam P. Sulochana Garu, respected Patron, Managing Director P. Pranit sir, and today our most valuable resource person, Professor R.Nagaraju sir, Dean of Sciences, Pharmacokinetic University, Tirupati. and all my fellow colleagues, the Department of Pharmaceutics and other allied departments. Very good morning to all. And I furtherly wish a very good morning, warm welcome to all delegates who are participating in this 2 days webinar program. The main concept to entertain in the lockdown to pandemic stage, this is the way the opportunity where we can update ourselves and we can clear that no critical gap between this research and the academic area. In part of this, we took this 2 days national workshop, the concept of pharmaceutical sciences and their principles and application. Today, Prof.R.Nagaraju has considered our request and he has the first resource person for the today's session.
Sir, excuse me, sir.
For this kind consent, I convey my wholehearted thanks on behalf of my institution and on behalf of my management. I welcome Professor Nagaraju for today's section. As you know very much aware, this today topic is application of pharmacokinetic principles in the clinical situation. As you are very aware, without application of pharmaceutical kinetics, we cannot ensure the validation of therapeutic values. Especially, it's well known to our industry area and R&D it has been enriched with this all pharmacokinetic applications. And secondly, it has had that kind of versatile phenomena in the field of preclinical studies and in vitro studies. It's very much need for the present scenario, especially not only the industry oriented, it will be required in the implementation of preclinical and other the clinical studies of different pharmaceutical validation. In the such regard, I requested Professor Nagaraju to deliver this appropriate topic for the need of research-oriented people, and for most of D people are quite used to this kind of program. I hope definitely it's going to enrich the minds of all youngsters of both PharmD Doctor Pharmacy program and M.Pharm and research students. So with this junction, I remain here. I convey once again thanks for his really acceptance to deliver the eventual topic of this pharmacokinetic principles in the clinical situation. Thank you very much, sir. Professor Nagaraju.
Thank you, sir. Thank you very much for your
So,
sir. Sir. Now it's time to start today's webinar by Professor R.Nagaraju. But before starting today's webinar, let us have a brief citation of today's honorable speaker and eminent personality in the field of pharmaceutical science, Professor R. Nagaraju. Now I request Srimati P. Kirtisika Madam to present brief citation of today's speaker, Nagaraju.
Good morning, sir. Good morning to our respected and honorable speaker, Professor R. Nagaraju, sir. I deem it a great privilege to present the profile of an esteemed personality, Professor R. Nagaraju, sir, Dean of Sciences, Sri Padmavati Mahila Vishwavidyalayam, Tirupati. Sir completed his B Pharmacy, M Pharmacy, and his doctoral degree from the College of Pharmaceutical Sciences, Manipal. Sir joined in Vishweshwara Institute of Pharmaceutical Sciences, Bangalore on December 1986 and worked at various professional positions like assistant professor, professor, head of the department, and contributed for the establishment of the department as well as the institution. Sir later joined KLU Society College of Pharmacy, Bangalore, and also during the tenure in Bangalore, sir worked at various positions in the university such as BOS member, expert committee member, inspection committee member. And sir served as member of organizing committee of various conferences and seminars organized by APTA, IPCA, etc. So coming to the prestigious awards, Sir has gained many awards. So I am very much privileged to announce the awards gained by Professor R. Nagaraju, Sir. Sir received the Best Pharmacy Teacher for the year 2018 by APTA. Sir was a recipient of Best Teacher Award 2017 by the Government of Andhra Pradesh. Sir received AP Scientist Award 17 by Andhra Pradesh State Council for Science and Technology, and also National Best Teacher Award by CVS Krishnamurthy Charities Tirupati. And also, Sir received many awards like Best Teaching Pharmacist Award 2017 by Indian Pharmaceutical Association Tirupati, and also many Scientific and Innovative Scientist Awards by IPA Anantapur Local Branch. And Sir was recipient of Distinguished Alumni Award of Manipal College of Pharmaceutical Sciences, Manipal for the year 2005. And Sir was recipient of the Achiever of Year 2002 award by Avopa Bangalore. And Sir secured Inform University first rank. And Sir was secured 2 patents on dental implants. Sir currently working as Dean and Professor at Sri Padmavati Mahila Vishwavidyalayam, and during his tenure at the university, Sir has held many positions like head of M.Pharm Pharmaceutics Specialization, and Sir started M.Pharm Pharmaceutics Specialization and established the laboratory setup on par with the premier organizations. Sir was responsible for establishing separate animal house with all facilities as per CPEC norms, and the other 2 specializations such as pharmacology and chemistry, which were started, were also given to the attention, and laboratory setup was strengthened under the guidance of Sir. And Sir was responsible for filling many vacant positions in teaching during the period as head, and effectively projected the institutional achievements visited by High Power Committee. And Sir worked as coordinator for the first ever UGC-sponsored 3-week refresher course on advances in pharmaceutical education and research organized by IPT. And Sir worked as coordinator for the UGC national seminars, and Sir has given the good leadership in getting the approvals from PC and AACT to the institution. And Sir worked as BOS chairperson or member at the university. As a BOS member, several initiatives were taken for the effective improvement of teaching learning process. Sir was responsible for introducing the interdisciplinary subjects at PG level, and also served as the BOS of many universities like Sri Krishnadevaraya University, Anantapur, Kaktiya University, Warangal, JNTU, Hyderabad, and JNTU, Anantapur. As a first BOS chairperson of College of Pharmacy, Sri SK University, Anantapur, Sir developed the curriculum for B.Pharm and M.Pharm. So, Sir received Best Teacher Award by the Government of AP, and nominated as MSET Committee Member from the year 2007, and 14 nominated as Executive Committee Member for APTA Central Committee for the year 2008 to 2011. So, Sir has 20 years of research expertise and guided more than 38 students and 8 PhD students. And Sir, under the guidance of Sir, many research projects were carried out. DST Curie 2-phase project of rupees 22.5 lakh was sanctioned, UGC project and AACT MODROPS project. And Sir has published more than 55 research papers under national and international, and Sir gained 2 patents, and Sir worked as member on the editorial board of 4 referred journals. Sir organized many seminars, conferences, and symposias. And as a guest lecturer, Sir attended many conferences and presented papers and keynote addresses, and contributed as chairperson and member of scientific committees of various conferences. So, Sir is a life member of IPA and life member of APTI, and Sir worked as Honorable Secretary of APTI branch. And Sir, held many administrative positions like Dean Development Chairperson, Coordinator, Director, Center for University, Member of R&D Committee, Member of Standing Council, Head Institute of Pharmaceutical Technology, and Chairperson. Sir, it's really a great honor and privilege to present the profile of you, and thank you once again, Sir, for accepting our invitation and for being with us today. Once again, welcome to the webinar, Sir.
Thank you.
Thank you, Kirtisika Madam, for giving such an citation for eminent personality. Sir Nagaraj, sir, shall we start session, sir?
Yes. I'm ready. Shall I start?
Yes, sir. Yes, sir. I request the Honorable Speaker Nagaraj, sir, to start today's session, sir.
Yeah, I am starting the sharing the screen.
Yes, sir.
Are you able to see the slide?
Sir, no, sir.
file. I'll come back again.
Yes.
Share. Yeah, now?
No, sir. Wait, sir. Wait, sir. Now it's visible, sir.
Visible. My voice is
Yes,
clear?
sir. Yes, sir. It's clear, sir.
Yeah.
Yes.
Yeah. At the outset, very good morning to all of you. Respected pattern and chief pattern, Sulochana madam and Pranit garu, and my dear friend Ranganayakulu garu, and my dear colleagues and colleagues around the SPSP. A very good morning to all of you.
Good morning.
And also, I also appreciate the initiatives taken by Sri Padmavati School of Pharmacy to organize in the pandemic situation, enlightening the delegates about various advanced aspects. And I get to understand that there are around 250 participants across the country. There are more than good number of participants are available even from outside the state as well as the different part of it. That is the technology advantage what we have taken. And I also thank, I should say, doctor, maybe she will become doctor now in another 3-4 days. So Kritisika for her elaborating
Thank you, sir.
of my biodata should not have been so long anyway. So I thank each one of you for giving me this opportunity to interact around 250 people. So I take it this as an opportunity, not as a privilege, because I am able to reach out around 250 people around the country. That is really a great thing. This platform provided by Sri Padmavati School of Pharmacy is very much, now I am very much delighted to have this type of platform to share my thoughts. Thanks again to the Padmavati School of Pharmacy. Coming to the similarities of our colleges, both Padmavati School of Pharmacy and Padmavati Mahila Vishwavidyalaya for Pharmacy, we started in the same year, completed almost 25 years, and still people have the confusion, Padmavati School of Pharmacy and Padmavati Pharmacy Mahila University. So that is the way the things people are. There are many students from the MSET will come to Padmavati School. Students to ours and our students come to your school. But anyway, not only the name, the fame and also the legacy what your institution is under the leadership of Madam Pranit and Ranganayakulu garu is worth noting. I hope this should continue and this is going to become further the best institution in the state of Andhra Pradesh and also in the country. Yeah, let me come back to my topic of discussion. And organizers, if at any time I am not audible, my slides are not visible, please alert me so that I will do the justification what is going to be. Right?
Yes, sir. Yes, sir.
Yeah, thank you all. So the point, when Dr. Ranganayakulu asked me, Nagaraj garu, please give one lecture on biopharmaceutics, and I could not say no because biopharmaceutics and pharmacokinetics is a subject very, very nearer to my heart. And I thought a topic which is having a lot of calculations and a lot of graphs must be really challenging to make you to give what point I need to give it to you. So I not touching the basic concepts in the pharmacokinetics. So I am not touching the fundamental aspects of it. I am only talk about the applied part of it. So for the students who are not that much strong in the kinetics part of it, please catch it and go back and refer the fundamentals part of it. Then therefore and connect the things. Anyway, I will try to share these PPTs to the organizers and also maybe slide share. So therefore it will be helpful to you. Yeah. And as I was telling you just now, and even though the equations and the graphs are many, I may not read out the equations or I may not tell you, bore you of talking something which is already present on the screen. Probably I will try my level best to apply the knowledge what you have gained in pharmacokinetics. Yes. This is our university entrance arch, and we have our university, 23 courses are there in sciences, and we have around 5,000 students in the total university, and pharmacy has started in this university in 1995, and 25 years is over, and this is our silver jubilee year, and pharmacy institution has got constant recognition from NIRF. That is, we have got 42 rank last year in national level. That is, in the state level, we have number 2 position, and we are striving hard to not only emphasizing on teaching learning but also research. That is the advantage in the university, where you get the grants, you get the support from the various funding agencies like UGC, DST, SRB, and name all central government funding agencies will first prefer the universities to be funded. So I am fortunate enough to be identified by the university and could run projects of few lakhs, approximately 2.5 crores projects has been sanctioned, and I have successfully executed all of them. So that is an opportunity that is only present in the universities and deemed universities to some extent. So that opportunity of the students also have got the exposure of such an what I should say, exposure to the researchers and also the end staff, and a blend of senior and junior faculty, and also develop the interest towards the research. That is very, very important. Reading the 4 lines for the what as per the syllabus of PCI is not enough. Those students, if at all the research scholars and other people are there, please ignore. This is only for students who are on the line. And the 4 lines of syllabus is not enough. You need to know more than that, and probably the environment makes you to learn more. Unfortunately, we are for last 2 years we are having a lot of problems in especially teaching and sector, where we are not able to deliver the things better to the students way. Anyway, this technology has made us to pierce in such ray of hope. Therefore, you can physically need not come to Tirupati, and whereas we are reaching you, that is really excellent opportunity given to by technology to us. And as I was talking to you, reading from a syllabus is not enough. You need to go. You need to have 5 important characteristic features to develop. That is a credibility, commitment, quality, transparency, and the trust. These are all the 5 inbuilt characteristic features you need to develop during your course study. So that as an end 18 years old guy or girl, you are going to enter intermediate, 14, 16, yeah, 17 years approximately, you are going to enter the college. And when you come out, you are going to come out as a PG, say 23 years, almost 4 plus 2 years, you are going to be in the campus. This 4 to 2 years, 4 plus 2 years is a very crucial life direction of your this thing you may be able to make. So there you are going to develop these qualities. Till then, till intermediate, you have been forced by your parents or the teachers to study, reproduce, regurgitate, and mug and do other things. So, but in the professional course, that is not the course. That is not going to be the thing. So you need to know. You need to have your what you want to go, what you want to be down the line after 6 years or 4 years. So that is going to be very important, and that only makes a way for you to foundation to move ahead. So these are the qualities you have to require in you when you progress from your adolescent age to the adult age. When you come out of the university or the college, you are grown up and you are able to take decisions which are going to be very useful for your future. That is very, very important. And all of you must understand that we have a lot of turmoil in the job sector, and we need to not only make your skills such a way that you are going to do the job, as well as do some innovation and some creativity or something where you can start your own entrepreneurship. So that should be the focus in future course of time. So many a times we tell this hard work is very, very important, and also the attitude of yours. So the hard work, and nothing will come simply like that to us. Going at 10 o'clock, coming back at 5 o'clock will not fetch you anything. It is going to be your constant work where you are going to do from the day one. So when you see it has taken 25 years of time for me to myself convince that I am eligible for some recognition in the field of pharmacy. So it is not come one day; it has come from the day one, and your commitment and hard work is very, very important. And the hard work not only give at the end of the day very sweet returns, but also smart also. You should be very smart, and you will get very smart and faster returns. So as I tell you, for a success, there is no shortcut. There is no shortcut for success. Don't compare yourself to your software people. Don't compare yourself with medical people. Their professions and their job profile is totally different. So what justification you do for your work, for your pharmacy, and how you are going to come out where you are able to successful, that matters. So that is very, very important. And whatsoever you have to do, it should be like perfection. So as I tell you, in many, you enter in the university or the college at very tender age and you don't know anything. The first thing is you will be freed out of all the clutches of your parents. You are very, very free. You are going to have a smart mobile, you are going to have a bike or so. You have no restrictions at home, and people will not talk to you 10 to 9 morning, 8 to night 6 o'clock. You will not have any study. It's all only, everything depends on you. So you need to cultivate the reading and skill thing and achieve the perfection. It is not simply one day reading before and getting 18 marks is not enough. So 18 marks anybody will give, anybody can get. The first class of JNTU, first class of Mahila University, anybody can get. So what is different in you apart from that? So what extra apart from the subject? So what is the real-time situation you are handling? How all this is going to be a matter? And from the students' point, from the line of the students, I think you need to go into that direction. And many a times, the environment where you study matters a lot. And when you see all India level, the syllabus of Pharmacy Council of India prescribed the same, then why students from different institutions are different? It is not. It is first is yourself. You are important as another student, and next is your institution where you are attached. So these two are the both sides of the coin where you are going to your success is going to matter. So the good principles of institution will help you in making featuring your thing, and your attitude towards the perfection will further strengthen. And you should not stop living. My teacher didn't teach this topic. Why I should study? I am already got 18 marks. You study for your knowledge, study for your understanding, study for your future. So that is important, not for JNTU degree. So the perfection and every step what you do, you should be able to lead to the perfection. Even I am retiring another 2 to 3 years. Even now, yesterday you ask Professor Raghunathan and his team. Yesterday 7:30 we had a training session for me. I only opted for this. I want because this StreamYard is not known to me, and I told Dr. Shanmugam and his team to help me out in learning these things. So yesterday I spent around half an hour on this, how I learn this. But however, in the morning we had some technical issues. So what I mean to say is learning should never stop. Learning should be continuous, and acquiring knowledge is going to be continuous, and that is going to be that attitude you should have. And gone are the days where the importance was given only for the degrees. And what degree you have got, that is taken as an important. Yeah, you have got the B.Pharm degree, okay, give you this job. You have that degree, you have this thing, you get a government job. So the days are gone now. The days are now, there is a shrinkage in the government jobs where it is comfortable, people think. But I see no difference between government and private, maybe a little bit of job security. Other than that, no much that advantages are there, except a little pay and the pay perks, and as well as the, I'm talking about the teaching. So the teaching jobs are very, very small. So you have pharmacists, you have enormous opportunities in other sectors. So for everything, we have the competition. You have the people with B.Pharm degree. So many people are there. When we advertised last time for the data entry people, you don't believe in university. We received 250 applications from not only from the intermediate to the MTech. So we conduct a screening test and shortlisted 20 of them, and we called for the interview. So even out of 20, we are not happy with not happy with only 3 to 4 people, and that is what it speaks about. So it is going to be your responsibility to build in the quality of your learning and the quality of your work, what you are going to do. That should be very, very appreciated, and future that is only will help you, and things are going to become all right only in that. So for that, you require the hand holding from the staff, and you should have a continuous interaction with the staff, and have the self reading. Now we have so many online reading materials are available, online educational resources you call as. So you have many things are available. You are not being stopped not to do that. You can do read that, and you can watch that, and read the textbooks and do all these things. And since we are not able to give you a perfect, 100% practical training, probably that angle we need to strengthen in case you are going to come to the campus. And not like before, you have many options in front of you depending upon your interest and things. And not only the pharmacy person should go to only production. That was the thing before our days. So only the industry, some opportunities were there. Now you have many opportunities. You have clinical data writing, and you have pharmacovigilance. Name, so many are there, and therefore you need to choose which option you need to, and prepare yourself from the ground level itself, so that when you complete 4 years or 6 years in our life, you are able to come out with a clear goal. So you should not think that, okay, I will do after my 4 years, let me think what I should do, or say after my inform, let me think what I should do. No, that should not be, that is not the correct attitude. You should start by lettering in the course itself what is going to be your ambition. I have many students in the university and in the private college they work. The very first year, some students are very different. And when I asked them what is it, they said, "Sir, I want to go to US." So that is what the first ambition in their life. So for to go to US, what are the things they have to do? They start doing very first year. And some students say, "Sir, I have studied BPharm in this. I want to do MPharm in some best organization, say like NIPER or Manipal or Padmavati School or whatsoever it is going to be." So for that, they pick up the way of life. So that is required for you. So you need to choose your line of interest, and maybe in first year and second year, and go along with it and strongly believe in it. And some students will say strongly, "Yes, sir, I am interested in teaching. I want to do in teaching. I am interested in research. So let me go to the research." Now you have plenty of opportunities to work as a research pharmacist. So likewise, you need to design your life in such a way that you are going to be successful. And the most important satisfying word is the we, the teamwork. That is very, very important. Please remember, nothing will happen in this world because alone. You see from the morning how to make this program successful, there are almost 5 to 6 people are working in the background. The whole team. If even one person, some problem is that the whole show is going to go waste. So that is, you need to learn and keep aside your ego, and try to be humble and see how you are going to work. This is applicable to everyone. This is applicable to everyone, from the teachers to students also. And learn from the beginning how to work in a team or how to lead a team. So both are important. So it is going to be we. And when we had some success in the thing, we always said, "Yeah, I am because of me, because of Nagaraj, this is success." No, we should be, even though that is because of me, I should not say that. You see, it is a teamwork. That is a humble thing you need to have, and you need to develop, as I said just now, the teamwork capability and also leading a team. So there are many people in the student life itself identified as a good team people. Yeah, when you give some event, some students extraordinarily meticulously plan and do it. And when you start something, immediately they have a proactive. You can see in your group itself. I think the N faculty in SPSP agree with me. Some students have leadership qualities and we can easily identify them. So that does not mean that don't think that leaders are born. Leaders are made. So it is how you are going to select your thing. That is not in the what I want. If I have to organize this program, what I will get. Instead, I will go to a movie and sit and actually see. Or instead of that, I will see I have mobile, I have some movie I have to see. I have some WhatsApp I will do. So if you have that particular attitude, probably it is going to be very, very difficult in future. And at this stage, what you are in a stage of receiving, and receiving will be successful only when you talk less. So make the other person who are in front of you to talk more. Therefore, you will get the more information from the that particular person. If you try to talk more and you will not know the other person, opposite person. So wherever you need to talk, you need to talk, but most of the time you need to have a patient listening. First, become a good listener, then you are going to be successful. And we know we can assess the in psychology. People say that a very smart person will not talk much, right? So they all will allow the other people to talk and get the feel of it, and they act very smartly. So this is what you need to, especially the youngsters, need to cultivate in your personality and transform you a person which is empowered at the end of your course, what you are going to come out. I hope I'll had enough introduction to you to the life. Now let me come to the topic of today's topic. And I am going to speak to you a brief about the set introduction. It is called an introduction to the topic. And the three important aspects of the application of the pharmacokinetics are TDM, individualization therapy, design of dosage forms. As just now in the introductory talk I have mentioned to you, there are so many graphs and equations. Don't worry about it. And those are available in all standard textbooks available. And you need to know how these are used to get your answer. So that is more important, and that's what I am going to be focusing. So you know very well, as I just now in the beginning I said, I am not going to talk to you about how to calculate pharmacokinetic parameters. But anyway, it is my small duty to refresh your mind about the pharmacokinetic parameters. So the pharmacokinetic parameters are those which are very important, are obtained. We have the primary pharmacokinetic parameters, we have secondary pharmacokinetic parameters, and these pharmacokinetic parameters are calculated based on the use in the drug developmental level, and those are very important for drug approval also. So we have the different methods to find out the pharmacokinetic parameters, like blood plasma concentration time profiles. We have urinary excretion data, and so on. We have different methods to find out the pharmacokinetic parameters and how these pharmacokinetic parameters are useful to us. So that's what is going to be my topic here to talk to you. So the important pharmacokinetic parameters I have listed on this particular slide. I hope it is clear. Volume of distribution, dose by concentration in the plasma genotype. Plasma concentration, plasma clearance, sorry, is equal to rate of elimination multiplied by volume of distribution. Plasma half-life, you can obtain directly from the graph, or you can calculate it, or it can be taken by the simple equation by 0.693 divided by the elimination. And the most important thing is bioavailability. So bioavailability, you have both absolute bioavailability and relative bioavailability. You have various methods to calculate it, and those parameters are going to be very significant in applied part of it. And what is the difference between pharmacokinetics and clinical pharmacokinetics? All of you know, pharmacokinetics is the study of time course of drug absorption, distribution, metabolism, and excretion. Time course here means what I mean here: rate of drug absorption, rate of drug distribution, rate of metabolism, and rate of excretion after the drug or the formulation is administered to a individual. So to study all these, that's why it is called as time course. Time course. So time course is a change in that any particular parameter with respect to time. So that you will be able to get the rate. So the clinical pharmacokinetics is pretty different, where it is going to be the application of pharmacokinetic principles to the safe and effective therapeutic management of drugs in an individual patient. So the clinical pharmacokinetics is going to be the applied application of pharmacokinetic principles in the individual patients. So usually clinical pharmacokinetics in the Indian setup, we may not be able to apply 100%. But anyway, these are going to be useful in therapeutic drug monitoring in case individual patient is going to be required. So the clinical pharmacokinetics principles are going to be basically categorized into three categories. Number one, therapeutic drug monitoring. Number two, clinical management of individual patients. See, the clinical management of individual patient is going to be a myth in our Indian conditions, and all of us are treated with the same dosage regimen. But whereas the pharmacokinetic principles are there even within channelize the individual patients how we are going to manage the condition. And the more importantly, if you want to design for any drug a suitable optimal dosage regimen, so these clinical pharmacokinetics principles are kinetic principles are important. So like if you have a drug A, how what dosage form it is going to be suitable, whether it is going to be an IV route, whether it is going, the IV injection, or it is going to be a tablet, or it is going to be capsule, or it is going to be any other dosage form, and to obtain a particular dosage regimen. So what needs to be done? That knowledge, the applications of pharmacokinetic principles we are going to get. Let us briefly talk about the first one, TDM, therapeutic drug monitoring. And many a times the students, especially the undergraduate students, we have some difficulty in pharmacokinetics, and because of the fear and the phobia, they try to leave some of the equations and the graphs, thinking that it is difficult. So it is not difficult. Learn it with an interest. Learn it with a challenging. Definitely, it is going to become a very important. And if this is going to be biopharmaceutics and pharmacokinetics topic, and the knowledge is important in every part of your pharmacy profession. So whether you are in industry or in R&D or in any other profession, the biopharmaceutics and pharmacokinetics principles are very, very, in one way or other, it is directly or indirectly related. So therefore, if students have got that phobia, please come out of the phobia and take the help of your teachers and your mentors or your seniors to overcome that phobia and try to learn it with a different angle after listening to me. Definitely, you enjoy it. So why I say this is, when I was a student like you, biopharmaceutics was in third year for us. In third year B Pharm means in 1983, I was in third year B Pharm. Many of you may not have born. So, and maybe your parents' age. So I had a really difficult time with understanding the pharmacokinetics part of it. And in first session I left, but teacher called me and said, "See, you are very good. Everything is very good for you. And why you are not attempting the pharmacokinetics? I expected you are going to attempt." I said, "Sir, I am very scared." And the teacher really helped me in understanding and motivated me. Therefore, the next sessional, I attempted only pharmacokinetics and got full marks on that. So when I can do it, the generation now is much more smarter, much more intelligent, much more, what I should say, everything compared to our generation. You can do it very easily compared to us. Right. So the therapeutic drug monitoring basically reflects about the individualization of the dosage. So the dosage need to be made individually to the patient by maintaining the plasma or blood drug concentration within the target range. So what is the target range? Target range is therapeutic range or therapeutic window. So you need to use the pharmacokinetic principles in such a way that how much has to be given initially, the loading dose, how much has to be given subsequent doses, and how these things has to be, how the drug concentration is maintained in that particular level. That's all going to be, you are going to talk with the therapeutic drug monitoring. So why therapeutic drug monitoring is very, very important. Even in India, few of the cases, the therapeutic drug monitoring is taken as a hospital management of the patients. So this reflects about the adherence of the therapy, whether how well the patients are taking the drugs what is being prescribed by just checking their blood levels, it is known. And more importantly, to individualize the therapy. So the individualization of therapy is very common abroad, especially hospitalized patients, and that is slowly coming in India also. We also use this TDM to diagnose the toxicity levels, whether the drug is going to toxic levels or not, and also use this TDM understanding to guide the withdrawal of the therapy. There are some drugs, very well, we suddenly cannot be withdrawn, especially the drugs like neuro drugs, which are going to be used acting on central nervous system. We are not supposed to withdraw. Or if, for example, we are using the steroids, immediately we are not supposed to withdraw the thing. Likewise, we have quite number of drugs and categories of drugs where the withdrawal of the therapy immediately is not advised. In such cases, TDM will help us in what way the withdrawal schedule can be made. And we also understand that whether the patient is already unknowingly or unknowingly taking the drug what is being prescribed by you before starting the therapy. So sometimes, say for example, we get admitted the unconscious patients with an asthma, and we don't know the attendants will not tell what happened, and the patient is asthma, and we want to know whether he has already taken theophylline or not. Because if it is a known asthmatic patient, probably he might have taken the theophylline. So quickly, by estimating the drug plasma concentration level, we'll be able to see whether already this particular patient is before coming to the hospital taking the drug. So that will help us in designing the dosage regimen for this particular patient. And of course, it is very much helpful in research and development to monitor the drug interactions in especially post-marketing conditions using the population kinetics. And all of you know very well the phase 4 clinical trials are very, very important. And the drug interaction by itself is a specialty chapter where most of the pharmacology students will read as one particular course, that means one subject. That much vast interaction principles are there. So therefore, you need to monitor in any such interactions, because all of you must agree with me. In India, physicians has got right prescribing the drugs a what I should say polypharmacy, maybe polypharmacy. That means more than two drugs. When you see any prescription, you can see the analysis of the prescription. Recently, one of my students, she wanted to do something else other than this. Is what we get some students request. Sir, I'm very free in our this thing. No much COVID is there. Can I do something sitting at home? Then I said, how many medical shops and hospitals do you have? Yeah, she said some number. So one student I gave this exercise, even though it is very known to all of you. I told ask her to collect 100 prescriptions with their permission. Like take a photograph, and after 100 prescriptions, you tell me what is going to be completed or not. So after 2 days, she called me promptly. Sir, from one medical shop, I sat 1 day or 2 days. She got 100 prescriptions and they are allowed. And I asked her to identify how many what do you call polypharmacy. It's not surprise to us. In more than 90%, we have the drugs are more than two. So this is called as a polypharmacy. In case such next step was, I asked her to see any drug interaction. Is no drug interaction is possible. She's working on it. So likewise, we need to have so many things are there which can be the student can think of and do innovatively. So therefore, the point of action point, what I look into here is the drug interaction, and the post-marketing surveillance, the pharmacokinetic principles are very significant. And this drug plasma concentration monitoring may not be highly possible except in few hospitals for the drugs which are having a low therapeutic index. The low therapeutic index of the drugs which are going to be having a narrow therapeutic between the minimum effective concentration and maximum toxic levels that is called as the therapeutic index of what they call therapeutic window, very small, especially for cardiac drugs. So such drugs where we see the concentration is to be monitored very, very closely. So that is going to be important. The plasma concentration monitoring is going to be very, very important in such cases. So, and these are the therapeutic drug monitoring also focuses on the drugs which are not metabolized to active metabolites. So we have the active metabolites, again a different story. So probably we need to have those ideas into that. And the concentration is not predictable from the dose. So where are the drugs where the plasma concentration cannot be predicted? In such cases, direct measurement of the drug plasma concentration is important. So the concentration relates to well either the therapeutic effect or toxic effect. Uh, uh, then this will be very helpful, and whereas the information is going to be useful in further course of treatment. Especially the drugs are taken in an overdose. So this, especially the toxic effects, probably estimating the plasma concentration will tell us how much of the drugs are, what is the concentration of the drug, and how it is going to be, and what is going to be the line of treatment. All those things are going to be very, very important. So that can be obtained from the measuring the drug plasma concentration monitoring for drugs. These are the, I try to give the examples of the drugs where monitoring is helpful. I quickly read out these things without spending much time. Aminoglycosides, phenytoin, theophylline, digoxin, I gave this example just now. Then the anticonvulsant drugs and tricyclic antidepressants and anti-arrhythmic drugs. So what I want to impress upon you is there are quite number of drugs where TDM is going to be monitoring of the plasma concentration is going to be very, very important. So what is going to be this functions of this therapeutic drug monitoring? What advantages we are going to get, and how this is going to be give us the improved treatment what we are going to talk about. So the TDM will be mainly helpful in selection of the drug, what type of the drug we are going to, and also the dosage regimen. What is dosage regimen? Dosage regimen is something where what is going to be the frequency of the drug to be administered, and what is the time, at what time intervals we are going to get, and how many times in a day it has to be taken for how many days. So all of you know about that dosage regimen. And also determine the need for the measuring of the serum drug concentration. In some of the cases, we don't require actually the serum drug measuring concentration. That also can be obtained by monitoring the drug concentration, and this is very helpful in assay for the drug concentration in biological fluids. So we need to know how much is going to be present after administration in the various biological fluids, and we are going to test not only the blood or the plasma or the serum or similar biological fluids. We have saliva, we have fluids like lacrimal fluids, and also we have urine, and we have all the biological fluids, whatever they are measurable, are you able to see or can be done. And then whatsoever the drug concentration profile you are going to get, and you will be able to do the pharmacokinetic evaluation and find out. I'm talking with respect to the drugs which are under development, right? So we need to know the pharmacokinetic evaluation for the drug concentration. And during the development stage, we after understanding designing the dosage form, sometimes readjustment of dosage is required. if it is required, probably this therapeutic drug monitoring is going to be helpful. And to monitor the serum drug concentration for highly toxic drugs, and which are going to be very, very significantly obtained from these TDM studies. And I was talking about the for crucial step in therapeutic drug monitoring is finding out the drug in the biological fluids. Probably majorly it is going to be in the blood the serum or similar fluids. It is a challenging task to find out such a small quantities in the blood. You must be knowing there are some many majority of the drugs will not be available in the blood in a higher concentration that can be detected by a spectrophotometry, maybe use UV or visible. Sometimes we have got these drugs, highly potent drugs. The dose itself is in few micrograms and less than a milligram. In such cases, what is important is the assay of the drug method, the method development. That's why bioanalytical method development has come into a picture. Lot many pharmacy graduates or researchers taking this challenge in developing the drug assay method for the drugs in biological fluids. So it is going to be a little different than that. So where we need to use not only the different extraction methods from the drug and use the advanced equipment in the laboratory that need to be available here. We require invariably the HPLC is a very important tool, that is the instrument that is required for assay the drug at very less concentration. And the gas chromatography, nowadays the gas chromatography is not that commonly used, and instead they are going to go for HPLC with interface and something LCMS they are going to go for instead of gas chromatography. And we also do at a spectrophotometry to a what I should say level of UV level where some drugs can be detected in that cone, and fluorimetry in specific cases, and some specific cases you need to go for immunoassays and also use radioisotopes. So these are all some of the very important techniques where we can use for finding out the drug concentration in the plasma when the concentration is very, very small. So where the pharmacokinetic principles are applied and done using these things, and that is about the therapeutic drug monitoring. Next point is designing of the dosage regimen. So the dosage regimen is one. The designing are going to be, before coming to the market, the manufacturer should be able to, with the relevant documents, they have to submit this date. So the dosage form, dosage regimen is just now I mentioned to you. It is the frequency of administration of the drug in particular dose called as dosage regimen. Dr. Shanmugam, everything is all right? The people in the studio, is it all right? Everything is all right? Please confirm before proceeding. Srinivas Murthy or anybody? Okay, I think there is no response from their side. I deem it as everything is all right and proceed next. So the frequency of the administration of the drug in particular dose is called as a dosage regimen. So an optimal multiple dosage regimen is one in which the drug is administered in suitable doses which has been decided before with sufficient frequency. That means maybe 3 times a day or 4 times a day or hourly or once in a day or whatsoever it is going to be. That is the frequency. That ensures the maintenance of the plasma drug concentration within the therapeutic window for the entire duration of the therapy. So the entire duration of the therapy, the objective is the plasma concentration should be maintained just above the therapeutic window, just within the therapeutic window, just above the minimum effective concentration. So that is important. So to achieve that particular objective, we need to decide about the dose at which need to be administered at different frequency where we are going to achieve this. Then next is individualized dosage regimen. What is the meaning of this? This is actually the most accurate approach to a dosage regimen design would be the calculation of the dose. That is very, very important based on the pharmacokinetics of the drug in the patient. So to arrive a particular dose, we need to take the help of pharmacokinetic parameters. Therefore, how much dose is required for a particular patient? So, if the patient is admitted with a problem of, say, kidney problem or say liver problem, if the drug is mainly metabolized in the liver, if the liver patient is there, probably the calculation of the dose is important, because if you give the same dose as a normal individual, probably you are going to enter into the toxic levels. Similarly, in case of the patient with kidney disease and the drug is mainly excreted through kidneys, then when you're not adjusting the dose and you're not calculating the proper dose, the patient accumulates the drug in the body, the blood, and therefore may lead to toxic reactions. So whatsoever is going to be the individualized dosage regimen, this is going to be approachable, it is going to be useful. And please remember, this individualized dosage regimen is not feasible for the calculation of the individual dose. So individual dose is calculated separately, and that is already existed, and to make it further trimmed to the patient needs, this particular approach is going to be taken into account. We are based on the population averages, the dosage regimen is being fixed based on two models. That is a calculated dose based on average pharmacokinetic parameters obtained from the clinical studies. One model is the fixed model that assumes the population average pharmacokinetic parameters may be used directly to calculate the dosage regimen of the patient without any alteration. This is a fixed model, self-explanatory. Adaptive model is just now what I just told to you. A dosage regimen calculation uses the patient variables and known patient pathophysiological conditions. So the adaptive model is mainly focused on taking into the patient condition and what is the variables, and if the patient is a geriatric patient, and what is the problem. If the patient is a pediatric patient, what should be the dose? And the pediatric patient, and say you have got an adolescent patient, how it is going to be. And a person is an obese patient, how that dose should be. If the person is a very thin and malnourished patient, what should be. Those are all the patient variables. Whereas patient pathophysiology means it is the condition of the disease condition of the patient which is going to be taken into account. Just now I mentioned two examples, liver disease and the kidney disease. Likewise, different disease conditions will also affect the dosage schedule. So that is model is called as the adaptive model. And there are some models, regimens which are based on partial pharmacokinetic parameters. So we are not going to use completely the parameters of pharmacokinetic parameters. What is going to be. So sometimes we are going to get for the drugs where we don't get any complete pharmacokinetic parameters are not known, are not available. In such cases, we are going to use a different methods. So we are for this purpose we are going to take three important calculations for the account, that is the nature of the drug, safety, efficacy, and therapeutic range. So these three parameters are taken into account for calculation of the dosage regimen. So coming to the next important one, how do you determine dosing rate and the dosing interval and the maintenance dose? So these are all the three important parameters which need to be evolved based on the pharmacokinetic parameters. So if the given dose is CSS, steady state, and if we get CSS, if you get clearance, if you know T-half, it is possible to calculate these parameters. So subsequently in my presentation, I will give an example how to calculate this. So step one is dosing rate. that is R, that is a mass per time. Calculate that. Next is step 2, dosing interval H need to be calculated. Step 3, maintenance dose is to be calculated. You have 3 parameters in your account. CSS, CL and T-half is there, and we need to calculate these parameters. Let us see how these parameters are calculated. When you have CSS, a 3 microgram per ml, and CL 2.8 liters per hour, T-half is 10 hours. This is the data you have. To find out the step 1 dosing rate, use the equation R is equal to CSS into CL. So you will get the dosing rate is 8.4 milligrams per hour. So this is the first parameter you can calculate from the given data. Next parameter is how do you calculate dosing intervals? So we already know CSS, CL, and T-half. So what should be the reasonable interval of the drug administration? Usually at the end of the half-life. All of you must agree with me. After the half-life, when administered next drug, the dosage frequency will be good. So that means the H can be or approximately equal to half-life. Since half-life is 10 hours, both 8 and 12 hours could be considered, maybe 2 hours less and 2 hours later. So it can be considered as the next administering the next dose, that is a dosing interval. That is number 2 parameter. Number 3 parameter is determine the maintenance dose. So to get the maintenance dose, we have got the parameters. Already the parameters are CS, CL and T-half is there. And you know R and H, which are calculated from the previous calculation, and you can calculate the maintenance dose is equal to R into H. So that is nothing but 100 milligrams quarterly, that means 12 hours interval. So that is going to be to be taken at this. These are with the simple pharmacokinetic data, it is possible to calculate 3 important parameters that is going to be used for the maintenance of the patient's clinical maintenance, clinical situations. This example will be helpful, and you can use this for the further calculations and use it. When you see the curves, I was just now mentioning to you, you have, by the therapeutic drug monitoring, you have a plasma concentration and the time curves are there. And when you see the old one, which is the red, in the red, the minimum minimum effective concentration is given C, and minimum toxic levels is given the red. So you are going to get a red type of plasma concentration time profiles. That is not actually required for us. That is actually after the 3rd hour or 4th hour, it is going to go beyond the therapeutic window. So in such cases, we should have the dosage regimen which is between MEC and maximum safe concentration, that is MSC, between 6 and 20, say for example. So the blue one is the ideal one, where the blue one at the is going to be the required the dosage regimen change actually required, and if that is there, that is going to be very useful. So this is what you can calculate the dosage new and with the dose old and the concentration desired, concentration observed. So you will be able to get the next dosage regimen. So likewise, loading dose and maintenance dose can be calculated using the pharmacokinetic parameters, and this is what the equation you are going to use for this purpose. Any standard textbooks will be able to give you this particular equations. And elimination half-lives are used to select the length of the drug and dosing intervals. That's what this particular graph shows. 5 minutes of process. So the elimination half-lives are, this is the first, the half-life is 3. This is what the plot you are going to get. But we need to adjust the half-life, dosage regimen where you are going to get the center color, the red color, that can be obtained. Similarly, the peak plasma concentration can be calculated by the simple equation. As I promised you, I don't go into detail about this. It's available everywhere, but only thing is equations are not interesting to read unless you solve right down. So the tough concentration is going to be obtained like with this particular equation. I'm going to skip this. I already spoke about the dosage adjustment in different people, renal failure and renal clearance and elderly, and all those. We have different equations for that based on the pharmacokinetic principles. So to summarize, today's class, today's discussion, I try to focus about how the pharmacokinetic principles are going to be effectively used in TDM and individualization of the therapy, and also in the designing of the dosage regimen in case of the people with renal failure, liver failure, liver disease, and also in the elderly patients, and also in the small children, in neonates, and the children, where we are going to use a design whatsoever the requirement we are going to, and effectively apply these pharmacokinetic principles in clinical situations for the better therapy. And these are all the references. And also I should also thank Professor Mannaji for also giving some inputs in this, making the slides. Now he is a retired professor of Annamalai University. Yeah. So with this, I'll say thank you to all of you. And as this picture shows, you are, you are very important to your mother. See the sacrifice of the mother, by and large the parents, where in spite of the heavy rain, the mother would prefer to drench but protect the child. So that is the mother. So love your parents, take care of your parents, and take care of yourself in this pandemic situation, and try to be positive with all things it is going to be, and see how this pandemic situation can be made an advantage to you. I thank one and all for your patient listening. I hope whatsoever time 1 hour I am supposed to give it to you, the small piece of information what is being known to me. Thanks for your patient listening, I believe, and also thank the organizers to make me to be the part of this program. Thank you very much.
Yes, sir. Thank you very much, sir.
Can I stop sharing now?
Yes, sir. Yes, sir.
Hello.
Yeah.
Sir, thank you very much, sir.
Hello.
Thank you.
Rangnaikul,
Hello.
sir, your voice is audible, sir. Sir, your voice is audible, sir. Ranganaikul, sir.
Okay. So thank you very much, Professor Nagaraju, sir. You are indebted both the technicality regarding the biopharmacokinetics, including your TDM, the cost. Okay.
Thank you, sir.
Thank you very much, Professor Nagaraju, sir. So you related the concept of biopharmacokinetics as well as the moral education for all the students in the clinical pharmacy. So it is tremendous emphasis in this application of biopharmaceutics in the clinical applications in the clinical pharmacy. It's an enormous lecture. Definitely, the implication of this pharmacokinetics enrich the safety margin of drugs by considering the drug dose and the concentration. Definitely, I hope everyone is going to be carry out the knowledge of this pharmacokinetics after listening to this session. Thank you very much, sir, for your enlightenment.
Thank
And
you.
I pass on this mic to R.Nagaraju, please.
Yes, sir.
And I
Thank you very
request the Organizing Secretary Shanmugam to hand over the token of appreciations to Nagaraj, sir.
Dr. Shanmugam, can I leave
Yes,
the
sir.
studio?
Thank you, sir. Thank you very
Thank
much,
you
sir.
very much once again. Please try to get the feedback and please give it to me
Mr.
a feedback.
Yes,
Mr.
sir.
Shanmugam.
Yes, sir. Sir, your voice is audible, sir.
Mr. Murthy?
Sir, your voice is audible, sir.
No, we have other formalities. Okay,
Yes, sir.
okay. Then you proceed other formalities, please.
Yes, sir. I request admin staff, Mr. Jamal Basha, to please present appreciation certificate for Nagaraj, sir. Yes, sir. Thank you. So, thank you for Professor Nagaraj, sir, for being with us and sharing his knowledge on the topic, pharmacokinetic principles in clinical situations. That's it, sir. Dear participants, you will receive your certificates by mail within 15 days. Thank you all for joining with us. Sir, shall we join for national anthem, sir? I request Jamal, sir, to please go with the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you, sir. We thank our management, Chairperson Srimati P. Sulochana Madam Garu, and Director Pranit sir, Principal sir Dr. D. Ranganayakul sir, for providing all the facilities for the conduction of a smooth webinar on this first session, sir. Thank you one and all for joining with us, and very soon, within a span of 10 minutes, we'll proceed with the second session of day one of this national webinar. Thank you all for joining. Thank you very much. Thank you, sir, Ranganayakul sir, and thank you, Dr. Shanmugam sir. Thank you all.
Thank you. Thank you.
Yes, sir.
So appreciate all the pharmaceutical sciences, especially pharmaceutics department. The